Yeah, I'm Frederik, and I will be presenting a little bit about my PhD project, which is titled Medical Genetics in the Greenlandic Population. And that led to these two papers to the right, and they are kind of the main references for everything that I will show today. So today I will show you how genetic architecture can have a big effect on medical genetics, and I will show you how precision medicine can, in some cases, be better in the Greenlandic population than in the European population. But to get there, we have to get through a little bit of introduction, then we'll talk about genetic architecture, and then I have two examples in the consequence of this for medical genetics. All right. So Greenland is a very large country with a very small population of around 56,000 individuals. The current Greenlandic population is of admixed genetic ancestry, and that means that on average, an individual will have approximately 75% Greenlandic Inuit genetic ancestry and 25% European genetic ancestry. Today I will be talking about analysis from what's called befolkningsundersøgelsen, which is a public health survey in Greenland commissioned by the government of Greenland. And that obviously means it's a very big collaboration with a lot of different institutions involved. And I'm all the way down in the bottom here. I'm from University of Copenhagen under these three different professors. And this is just to say it's a very big collaboration and there's a lot of stuff going on here. And this is just a small part of it. So what are the public health surveys? The public health surveys has two aims. One of the aims is to measure the health in individuals. So they have this boat that travels around to the different smaller villages where there's no doctors or hospitals. And then if a person has something that should be noted, then they are referred to a doctor or the hospital. And obviously, if they are in a bigger city with a hospital, they don't use the boat. Then they also measure the health in the population as such, and these measurements are used to create reports that are given back to the Greenlandic government and used to inform healthcare decisions. And they have been going on for more than 25 years, and there's a lot of people involved. Today, I will only show you analysis that involve genetic data from these surveys. But why should we even study genetics in Greenland? And in my opinion, there's two reasons for this. One of them is that we know that genetics or your DNA affects diseases, most or many diseases at least, and in various different ways. And we also know that different human populations have different distribution of genetics. And that means we need to have a lot of basic research in medical genetics and also population genetics to eventually move towards being able to use genetics to offer better diagnosis and treatment of disease, which is what we term precision medicine. The issue is that most of the research that has been carried out up until now at least, has been focused on European or Asian populations. And that means we don't really know whether this also works for other populations. And this results in what we call inequity in genetic-based healthcare. Inequity is kind of a buzzword somehow, so I want to give you an example in our population of how that actually looks. So the example I will be giving is in the first-line diagnostic tool for diabetes, which is the HbA1c test. And for individuals with European genetic ancestry, this is a really good tool, and patients are diagnosed correctly and treated properly for diabetes. But if you're a person with Inuit genetic ancestry, then the HbA1c test underdiagnoses diabetes due to genetics, and that means that patients are left with untreated diabetes, and this exactly is an exam of inequity in healthcare, right? But in 2014, as I recall it, one of my supervisors, and in these public health surveys, they did a lot of research in the diagnosis of diabetes and changed the clinical practice. And this means that now they are diagnosed correctly, and this is at least more equitable than before. And this leads us directly to the aims of this talk. The first aim is to compare the genetic architecture of different human populations, and after that investigate the consequences from this genetic architecture for medical genetics. This is a simplified map of the Arctic regions. We have Greenland all the way over to the right. This is the Americas, Northern Americas, and this is Siberia. And in red, you see a very simplified schematic version of the human migration routes that different ancestral populations have taken during more than 23,000 years or something like that. And what you can see is that there's a lot of migration. All that's happening right here is quite complex, and that's not my area of expertise. But what we know is that around 1,000 years ago, the ancestral population that became the current Greenlandic population entered the northern Greenland part here. And the important thing from this is that we have a lot of migrations, as you can see here. And every time a population migrate, we know from a theoretical perspective that this creates what we call population bottlenecks. And we won't go into detail with that today, but the two main points you have to take home from these population bottlenecks is that one, most rare gene variants get lost, and two, we have few rare gene variants that become common. So we have fewer variants, but we have more common variants. All right. Then we wanted to see how it actually looked in the data from the Greenlandic population. So we whole genome sequenced 448 individuals from the public health surveys. And as far as I know, at least, this is more or less the first analysis of many whole genomes from Greenland. And then we compared to populations of the world. So first we wanted to look at the first statement, how many genetic variants do we have? And what you see here is the results from our sequencing data from the Greenlandic Inuit here. And up here you have the British, which is a European population, right? On the x-axis, you have the number of sequenced individuals. And on the y-axis, you have the number of SNPs or genetic variants. And what you can see, I think it's very clear, is that in Europe, we have relatively many genetic variants. And in Greenlandic Inuit, we have relatively few genetic variants, which exactly matches what we expected from these migrations in the ancestral population. All right, so the other thing was to look at how common the different variants are. And to do that, we here show on the x-axis what we call the minor allele frequency, which is a measure of how common a genetic variant is in a population. And on the y-axis, we have the proportion of genetic variants that are this common or more common. So that means, to take an example from a European population, the British one, we have around 43% of the variants that are common when we look at 190 individuals. And in the Greenlandic Inuit, we have 74% of the variants that are common. So in Europe, we have a relatively low proportion of common variants, and in the Greenlandic Inuit, we have a relatively large proportion of common variants. And that matches exactly our second expectation from these population bottlenecks. This is just to show you that this pattern is not only between Greenlandic Inuit and the European population. We also looked at Chinese population, African populations, and we see a similar pattern. I think it's important also to show that you can calculate something called the additive genetic burden in a population. And we see that across all the populations that we have tested, we have the same additive genetic burden, okay? But we can see differences within specific diseases, and we can see that there's a difference in the way the burden is distributed throughout the genetics. So that was our first aim. We wanted to compare the genetic architecture. We found fewer variants, but a larger proportion of common variants in the Greenlandic Inuit. And we see we have the same additive genetic burden across all the populations. And now we wanted to investigate the consequence of this genetic architecture for medical genetics. And to do this, I will give you two examples. The first example is for a complex disease. I will look at diabetes. And the second example is for what we call clinical screening. For the first example, we first start with the schematic of GWAS association results. And from a large population, such as the European, what you will see for diabetes or a complex disease in general is that our associations will fall into two main categories. To the very right here, we have the very common variants that have a very low effect. And that means even though a lot of people carry these variants, you can't really use these clinically, at least not a single of these variants. And if you go to the very left, we have another category, which is the ultra rare variants that have a high effect. And if we plug in the real data, then it looks like this, where we see that data from a European US study with 900,000 and an East Asian with 400,000 fall into these two categories. We have the common small effects, where we have the variant with the highest population impact, TTF7L2, and over here we have the ultra rare but high effect variants. To get these points here, we didn't even show you a single variant association. We had to group many of these predicted disruptive variants into what we call a gene burden. Otherwise, the points would be all the way to the left of the screen. And in this first whole genome sequencing study in Greenland, we identified this variant in a gene called HNF1 alpha. And I hope it's very clear that this is an outlier. It is both relatively common and has a very large effect. So this is a direct consequence of the genetic architecture, where we have a higher proportion of the common variants. We found this variant in around 7% of all the diabetes cases in Greenland. And if you're not working with diabetes yourself, we have to put this a little bit into perspective. To do this, we look at diabetes prevalence, and very roughly speaking, in Europe and in Greenland, the prevalence is around these 10%. And if we use these gene burdens that I showed you before with hundreds of variants grouped into a single gene burden, then we can identify less than 1% of the diabetes cases. And that's across hundreds of variants, so it's very difficult to utilize clinically. Whereas in Greenland, we have a single variant that's found in 7% of the diabetes cases. And that means we have a big potential for precision prognostics and treatment. Then we did a follow-up study, or it's in the same study, but a follow-up cohort, because as you can see, this variant is found in a gene called HNF1 alpha, and this is a relatively known gene that can cause a specific type of diabetes called MODY diabetes, and even a specific subtype of MODY diabetes, where one of the clinical features is that there is a low 30-minute insulin response to a glucose load. And so we wanted to test whether that was also true for individuals carrying this specific variant. So to do this, individuals were fasting for 12 hours, and then they were given a drink with a lot of sugar in it. It's called an oral glucose tolerance test. And then we measured different parts of the blood at different time points. And we are specifically interested in the insulin levels. And what we saw were at fasting, at the fasting time point 0 here, there's no difference between the individuals carrying the variant and the individuals not carrying the variant. Whereas in 30-minute time point, we see a quite big difference, a decrease in the insulin response to this sugar load. There was quite a big effect. It was also highly significant. And this tells us that it actually matches what we call the HNF1 alpha MODY phenotype. And what that means is that we can actually build upon the literature, what is known about treatment of HNF1 alpha MODY. And there's some different like preferred treatment tables where we can see that there's a kind of an old school drug for diabetes that could be used. So the hypothesis is right now that we could use this specific variant in a clinical setting, test that specific variant only, very cheap, very fast to do, and then we could directly give people the correct treatment and the correct diagnosis without them having to go through like different treatment regimes. So I think this is a super encouraging example of how we can actually use genetics to improve the treatment and diagnosis of disease. The second example I will give is an example for clinical screening, and I guess what Daniel talked about is kind of related to this, because you can have you can imagine that you have an undiagnosed patient with what we call a rare phenotype or a rare disease, and one of the tools a doctor has is to do a clinical sequencing of the genome. And then when you sequence the genome, you get thousands of variants out, and you have to figure out which of these variants could potentially be the causal one. One of the main tools we have to do this is simply filtering based on databases, because if a genetic variant is common in a population, it cannot cause a rare disease. So it's very simple. And then we end up hopefully with quite a very few variants. We might have some non-causal variants left, and hopefully we will also have the causal variant that we can use for diagnosis of disease. And these non-causal kind of false positive variants, the number of these is quite important, because a specialized clinician has to look through these variants manually and determine which one they believe is most probable to be the causal variant, right? Very simplistically speaking. So if we want to look at the number of these non-causal variants, then we can see for different populations here, like the British, a Chinese, and a Nigerian population, on average, we will have between 3 to 4 of these non-causal false positive variants, and that's a reasonable number for a clinician to look through. But as it is right now, in Greenland, the number is quite a bit higher, so there will be on average more than 12 of these false positive variants. And that means that they are currently disadvantaged, and we know from other studies in other populations that this actually matters quite a lot. It can lead to misdiagnosis and potential for health disparities. What I find super encouraging again is that using summary data from just a small number of individuals, we can reduce this inequity quite a bit. And as I said in the beginning, you can kind of think of it as this is actually a better estimate for finding the causal variant in a rare disease setting. So this is at least more equitable, and it's a lot better in Greenland with this summary data. All right. And so that was the second aim. I showed you that as a consequence of this genetic architecture, there's a large potential for precision medicine. And within the clinical screening setting, there's also a very big effect of inclusion. I want to conclude with this remark that I hope that I've showed you that there is this great promise for precision medicine in Greenland, but we have to kind of remember that healthcare is super complex and in many cases a political task. So there's many challenges in healthcare, and genetic-based healthcare is not always like the top priority. It's just, I think, important to say. So of course, thank you to all the study participants and the ethical committee and my supervisors and collaborators. And thank you for listening.
Yeah, awesome. Great talk, Frederik. Let's just first see if there are any questions in the Q&A. So there is one question. So besides looking at individual variants, did you also check for haplotypes?
We didn't look too much into the different haplotypes. The only thing we did with haplotypes was when we did our population structure analysis. We did that haplotype based, but we didn't look at the specific haplotype names and those kind of things. No.
I see. Okay, I'll ask one question. So you had mentioned that there were some rare genetic variants that became common in the Greenlandic population. So I was just wondering, when this genetic drift took place, is there a evolutionary reason or any comments on which are those rare genetic variants which became common and why so?
Yes, so there are multiple answers to that. I think the first one would be that there's a specific region that has been under positive selection, which has been shown before as well. So that's not, I'm not the first one to show that, in the metabolism of fatty acids. And that is most likely under positive selection due to the diet that the Arctic population had for thousands of years. But that's both true for the Greenlandic population, but also other Arctic populations that follow this migration route, right? And then we also show that there's no selection on any of the other variants that we know have an effect. So you could imagine that for some of the variants, it could be a lack of selection that allowed them to become very common, but we have not been able to show that statistically, at least.
Okay, cool. There is another question by Mariano. So, among the rare variants that you found in Greenlandic population, did you identify any genetic evidence of adaptation to the environment? If yes, are these variants also linked to some diseases?
I guess the only thing that we could prove statistically was the selection on the fatty acid variant. There were some other variants that have previously been suggested to be adaptation to cold environment, but we didn't at least see any evidence for that. And I would say we almost saw evidence against that because we saw a decrease in the frequency in Greenland compared to other Arctic populations.